Hi, this is Roger Green, host of the Surfing the Mesh Tsunami podcast. This week, we're offering 5 conversations from episode 20, our review of drug development stories from the EASL Congress 2024 with Jörn Schattenberg, Naim Alkhouri, and Mazen Noureddin. Plus, from the vault, one of our wrap-up conversations from the EASL Congress 2023. This wrap-up conversation covers a range of different developmental studies and goes on to a final question I ask. Naim, who has to leave early, lists 4 other topics he feels are worthy of discussion. Sagimet Bioscience's FASN inhibitor Denifenstat, Ionis's DGAT2 antisense inhibitor ION224, Takeda's TAK227, a transglutamase 2 inhibitor. Actually, it's Jörn who mentions that one. While all the group members believe that denifenstat's a good drug, there is some degree of optimism about how widely it will be used, with the most optimistic saying quite widely, others saying maybe a little more limited. Naim also discusses the result of SPECIAL, a study he participated in with the bariatric surgery group at Cleveland Clinic, evaluating the effect on the liver of bariatric surgery, specifically in patients with cirrhosis. The results were dramatic, with a 0.28 hazard ratio, or 72% reduction of risk for major adverse liver outcomes, and a 0.2 hazard, or 80% reduction of decompensation. As Naim points out, these are encouraging results, but they take place over a 10-year period, and it remains to be seen whether they are specific to surgery or can address any form of weight loss. After Naim exits, I ask Mazen and Naim what they consider likely to be the biggest stories at AASLD in November. Mazen opts for Essence, the phase 3 semaglutide study, while Jörn focuses on NITs. And with that, a really exciting and pietistic episode ends. During the episode, all of us kept comparing the negativity and virtual despondence of the 2019 to 2020 period described as the graveyard with what's described as today's gift shop of multiple modes of action, demonstrating clearly positive results. It's an exciting time for the field and for the patients. As I edited, I found myself thinking of Stephen's goal to put a big fat dent in fatty liver disease. We all can use this episode to gauge the size of Stephen's dent, and frankly, it's pretty damn big.
Naim Alkhouri.
So, Roger, I have to go. So let me just give you my spiel quickly on other mechanisms of action, and I'll let you guys debate. But as you mentioned, we have a couple exciting mechanisms of actions. Number one is denifenstat. This is a de novo lipogenesis inhibitor. Specifically, it inhibits an enzyme called fatty acid synthase. And what we saw at EASL was the FASCINATE-2 trial. So patients had biopsy after 52 weeks of treatment, and they hit on both MASH resolution and fibrosis regression. MASH resolution, if I remember correctly, MITT analysis was 36%, and then the fibrosis regression was around 40% with the 21% delta from placebo. So, an exciting mechanism of action, a new mechanism of action, and, they hit both the primary endpoints that we look at in MASH trials, the MASH resolution and fibrosis regression. I think there is a high discontinuation rate. These de novo lipogenesis inhibitors, there's concerns about hair loss and skin issues, especially with FASN inhibition. So we need to learn more about that. Going back to, the resmetirom data and just, comparing apples to apples. In my mind, I think efficacy-wise, similar to resmetirom, hitting on both endpoints is encouraging. But if resmetirom has the same efficacy and is better tolerated, then that's something to keep in mind. But again, interesting, different mechanism of action, and we know the disease is driven by different mechanisms, so it may work better for certain patients compared to resmetirom. The other mechanism of action was the Ionis DGAT2 inhibitor. This is interesting because, we're talking about RNA interference here, so I'm excited about that whole field, whether it's antisense oligonucleotides or short interfering RNA. But this was also a biopsy study, and they hit both MASH resolution. I think it was around 35%, and then fibrosis improvement. Delta from placebo was around 16% or so. Another, way to inhibit one of the de novo lipogenesis enzymes. This time it's DGAT2, but a different way to do this. So again, this is RNA interference. So very exciting. And then finally, I have to put a plug for myself, but I presented data on bariatric surgery in cirrhotics. This was what we call the SPECIAL study. This was done in collaboration with the bariatric surgery team at the Cleveland Clinic, where we identified 62 patients with MASH cirrhosis, biopsy proven at the time of surgery, and then closely matched them to a control group of patients that did not have surgery. And the primary endpoint was a percentage of patients that developed major adverse liver outcomes, MALO, so decompensation, HCC, needing liver transplant, transplant or overall mortality. And we looked at decompensating events. And to make a long story short, after a median follow-up of around 10 years, we showed a clear separation between the bariatric surgery cohort versus the medical cohort. The hazard ratio was 0.28, indicating 72% reduction in the risk of malo with bariatric surgery. And then when we looked at just decompensation, there was 80% reduction with bariatric surgery. I think this is important. We've shared similar data in F1 to F3 in the Splendor trial that we published in JAMA, but this one is in cirrhotics. And, there are concerns about the safety of bariatric surgery in a cirrhotic population. So it seems like in well-selected patients, bariatric surgery is not only safe, but it decreases liver outcomes. So that was encouraging. We'll see if it's bariatric surgery specific or is just weight loss and following them long enough. But again, being in a cirrhotic population was a big deal in my mind.
Jörn Schattenberg.
That was a great study.
Mazen Noureddin.
I just want to pick my favorite from what Naim said. A lot was said there. But Denifenstat, I'm saying correctly, Roger, but I think, to be honest, probably there was a little bit overshadowing because people like GLP-1s and duals and weight loss. Efruxifermin had a great fibrosis data at week 96. But with Denifenstat, I think people did not pay enough attention. I think they have a really, really good results, to be honest. To reemphasize the number, 41% fibrosis improvement compared to 18% placebo, statistically significant, and 38% in NASH resolution to 16% on the placebo. The MRI improvement was up to 65% compared to 21% with the placebo. I think it was a very well done study. It was intention to treat analysis. So they had similar, if not better, delta compared to many others that were presented. The hair thinning, I think, was transient, and people talking to the sponsor and being involved in their discussion, I think, is transient. The hair comes back. We'll see what happens in phase 3, but there are multiple strategies that are being implicated to mitigate this effect. Again, I think sometimes we hold other MOAs to a higher standards because they're not the GLP-1s that they have the great reputation of weight loss. So something to keep a good eye on. I think they have a very interesting product. They're going to phase 3, and I'm excited about this MOA.
Roger, as we're getting closer to the hour, I'm going to also mention one study I presented using transglutaminase inhibitor. So that concept is, again, a different mechanism of action. I think that was where the excitement of EASL comes from. We have so many different angles we can look at that people are actually having the discussion of what to mix and match. I think in the end, if we have a strong drug, we might not need to, but it's very interesting to see those multiple aspects we can target the disease. So transglutaminase does have the rationale of being used in NASLD for its anti-fibrotic activity and TGF-beta effect. Not a lot of clinical data in the liver field so far. It's been trialed in celiac disease for some other readouts here, but the early phase 2A trial I presented, and the substance is called EASL was 3 doses versus placebo after adjustment for the p-value negative primary endpoint. And that was a non-invasive test using ProC3, so no liver histology data. But the one thing, the concept that underlies that observation is if you look at patients with high ProC3, and again, that's potentially a marker that's elevated in patients that are more fibrogenic, so more fibrosing disease, you did see a dose response and a stronger decrease. And while not wanting to overpay a subgroup analysis plus hoc, I think the concept that certain patients that have actually fibrosing disease are more susceptible to antifibrotic mechanisms is very appealing to me. And it's difficult to favor in this type of activity in phase 3 trials, but I think in predefined subgroup analyses, you really get something out of that. If you look at the patients that are more likely to respond to your drug. We haven't seen too much of that, but I think we will more in the future.
Roger Green.
So as we roll to the bottom of the hour, a comment and then a closing question for you guys. The comment is that when I first started looking at this field 5 years ago, 2 things were true. Number one was everything that you were saying before, Mazen, about the drugs were failing and nobody was willing to invest. And the dominant question was, what do we have to do to get a 1-level reduction in fibrosis without even knowing exactly how we were going to do it? Obviously, this is a lot better. And my thought at the time was that this was like cancer, in that when Nixon declared the war on cancer in the first place back 55 years ago, the first thing we had to learn was that there's no such thing as cancer. There were lots of different ways to attack it depending upon where where in the body and what the genotyping was and all that other stuff. The idea that we've got 6, 7, 8 different modes of action all rolling through right now, one of the things it suggests to me is that if they all work 40% of the time or 50% of the time, I'll bet you it's not the same 50. And that there will be 2 challenges. Number 1 is getting things to the finish line. And then number 2 is once we get them there, understanding the disease at a level where we can go to the right drug first. And that should keep you guys going for your entire professional careers. But what it promises for the patients is even more exciting, which is we're looking at a place where in 5 years can be as exponentially better than now as now is from 5 years ago. And that would be pretty astonishing. With that, closing question. You guys are in a better position to answer this than me, so I'm going to just ask one big thing we're going to learn at AASLD, if you had a guess.
I think it might be the Essence trial, but I look for further analysis based on NIT from servotrizipatide. I think there's another FGF21 coming from Boston. I look forward to that presentation. I don't think PIMV will be ready for AASLD, maybe EASL, but maybe like Essence trial. Not sure if they will make it or not, but I look forward to that, which is the semaglutide phase.
Right. SEMA 3. Okay.
I'd say more companies coming to San Diego to put up their booth and actually get in touch with the physicians in the MASLD space. So while I cannot, foreshadow what's going to be selected or presented, I think we'll see more activity now with an expansion, interest, and that's reassuring for our patients. And then again, the big stories around NITs, we have only touched this briefly. We're going to see more data linking NITs to outcomes to finally also make a strong argument to use an NIT for conditional drug approval as it's relevant to patients' outcomes.
Yes, so Jörn, I'm looking, and we'll talk about this later, start pulling together an NIT episode for mid-July, which is the next time we've got free space exactly for that reason. The only thing I'd feel remiss if I didn't mention it at least once is, because Sven Franke did this in his follow-up interview with us last week, is no one's mentioned lanifibranor, which is just kind of out there trying to get a phase 3 started, of some fairly exciting phase 2 data, a different mode of action, and they're kind of in a lost in the sauce place in the process.
I wouldn't say lost.
They're always working in silence, and they come in surprise with good results at the end. Yeah. So, their phase 2 was impressive New England Journal paper, and they're working on their phase 3. So there's some people working in silence. So I look forward to that. Part of it is also, Roger, a lot of companies put data along the way with meetings. So that sometimes others get highlighted more than others. So we get excited with data and evidence-based medicine.
I just wanted to make sure they didn't get lost. That was all. I totally understand the reasons. So listen, let's make this a habit for future meetings, okay?
And now, back to Roger.
We hope you've enjoyed this recording. If you have any questions or comments about the content of the conversation or the entire episode, please put them in the reviews section of the page from which you downloaded this conversation, or send an email to questions@surfingmesh.com. Next week, Frank Taka and Luca Valenti from the EASD, EASL, EASO combination group will be joining us to discuss the updated clinical practice guidelines for Mazen. Until then, stay safe, surf on. We'll see you on the podcast. Bye-bye now.